Case Western Reserve University's Institute for the Science of Origins proudly presents the Origin Science Scholars Program. The Institute advances the scientific understanding and application of origins and evolution of human and natural systems. The Origin Science Scholars Lectures are presented with the assistance of Case Western Reserve University's Siegel Lifelong Learning Program, College of Arts and Sciences, and Media Vision. It's my pleasure today to welcome Doug Wallace, Director of the Center for Mitochondrial and Epigenetic Medicine at Children's Hospital of Philadelphia and Professor of Pathology and Laboratory Medicine at the University of Pennsylvania. Professor Wallace is one of the founders of mitochondrial medicine, and so it's a particular pleasure to have him here to tell us about mitochondria and their effects on humans.
Well, I really want to thank you all for the invitation and for you all for taking some time in the evening to visit with me. So we're going to talk about the human cell and an aspect of the human cell, which you heard something of from your lecture last week, the energy generating structure of the mitochondria. So the human cell, of course, shown here as a circular spherical cell. In the center there is the nucleus, which contains the vast majority of the DNA, but outside that are the structures which are in fact the mitochondria, and the mitochondria are symbiotic bacteria that live inside your cell, and they generate the vast majority of the energy that you use in your daily life. So sitting in your chair right now is 100 trillion separate individuals, and those separate individuals are cells. And those cells are interacting with each other, and by interacting with each other, they've created the illusion of a unity, and that is what you consider yourself. So that illusion of I-ness is in fact really a we. It's not an I, but a we. But interesting, inside each of the we is a they. And inside that they, there are literally hundreds to thousands of them in the they inside the we that is you. I think I got that right. But anyway, the point is that you are the product of two totally separate, independent lines of evolution. And those two lines came together, as you heard from Joe last week, in a fusion of these two life forms together to form this symbiosis, which then is what we call the eukaryotic cell, which you're seeing up there. So the eukaryotic cell has a nucleus and its cytoplasm. Well, that was one organism. That was an archaeobacteria at the time. And the other organism, these mitochondria, they are an oxidative bacteria called an alpha proteobacteria. But they were both bacteria, co-equal bacteria with the same size genomes. But over the period after the symbiosis, when they came together, they began to exchange genetic information. And so now many of the genes that were in the original oxidative bacteria have been transferred into the genome of the archaeobacteria to create this very complicated nucleus. But still, these are two independent life forms. So you are, in fact, a unique experiment in evolution. It is thought that this symbiosis only occurred one time. So all plants and animals, all complex things, fungi, yeast, they're all related to you through this one unique event. If that event had never occurred, there would only be very simple bacteria on this planet. And that one event transformed life as we know it. So this is a very important issue. Why would it then be obviously important, or why should it be obviously important? Because Newton said half a millennium ago that mass is inanimate unless it's acted on by energy. And yet you're the most animated thing in my environment, and I believe in yours too. So that means, since you are a mass, but you are animated, then the thing that's animating you is energy. And without energy, you would be inanimate, which is what we call dead. So wouldn't it be interesting from a medical point of view to understand the difference between being alive and dead? And that clearly must be energy. So therefore, the subject of today's discussion is the most important thing about biology. No biology exists without energy flow, and energy flow is why you are here. And the reason you can be complicated, why you can be 100 trillion cells, a colony, is because there's enough energy generated by these symbiotic bacteria to make that possible. The single cell bacteria cannot make enough energy to be complicated. But by creating this situation where you have one cell to harbor much of the DNA, but you can draw on hundreds of bacteria to make energy, then the sum of all that energy is enough energy to create arms, legs, eyes, heads, bodies, intestines. Okay? So this event, the creation of these energy factories, transformed life on this planet. And that is the evolution and the origin we want to talk about tonight. So, we're going to talk about evolution, and evolution has been associated in the last 115 years, well, no, actually, in the last 60 years, by the understanding of a molecule called DNA, deoxyribonucleic acid, DNA. But before that, a fellow named Charles Darwin, he had the idea that maybe, in fact, life evolved. That is, simpler forms gave rise to more complicated forms. And that idea of evolution was, of course, actively debated even to today, amazingly. But one of the things Darwin didn't understand is how the information occurred to create this evolutionary process. Now, this evolutionary process, we now know, is encoded by information molecules called DNA. So, at the turn of the century, of the previous century, the beginning of the 20th century, it was known from studies on fruit flies that, in fact, certain aspects of inheritance followed an inheritance pattern where it seemed like there were two copies of each inheritance factor, and then when the sex cells were made, one copy was put into each of the sex cells, and then at conception, the two copies came together to make a new individual. That idea fit very nicely with some observations a man named Mendel made in the 1800s, and it followed exactly with what the inheritance pattern of things of anatomical significance in fruit flies also followed. That is, if they had a bent wing, the bent wing was inherited in that way. Or if they had too many bristles on their bodies, or they had too few legs, they all followed this inheritance pattern. And that led then, in the beginning of the 20th century, to the idea that, in fact, genes were inherited according to the laws of Mendel. And then in the 1950s, DNA was discovered by Watson and Crick, and it was found that that followed that same inheritance pattern, and those DNA molecules were in the nucleus, and the nucleus had chromosomes where the DNA molecules were, and the chromosomal dance exactly followed the laws of Mendel. Fine. So that led everybody to say, wow, everything's solved. We now understand how inheritance occurs, therefore how variation would exist, and how by selection different variants would be forming, creating new forms. That's fine. That is called neo-Darwinism, and that is the fusion of classical Mendelian genetics and Darwinian evolutionary theory. But there are all kinds of problems with that theory. And I'm going to argue today that, in fact, there is a big disconnect between the idea of genetic variation in the nucleus and the ability for a group of organisms to adapt to a new environment and ultimately give rise to a species. And the question is, what allows that transition, that key transition of speciation to occur? And the answer to that question is going to come from very interesting discussion about what it means to feel badly. And so, our interest in our clinic is three things. Who are we, where do we come from, and why do we feel bad? And so we're going to try and figure that out in this discussion, and lo and behold, it's going to give us a new insight on how we all arose and the origin of species. Okay, so as I said, the cell is two life forms, and one life form now makes up the nucleus cytosol, and that life form became specialized in making anatomy. So all the genes for anatomy are now in the so-called nucleus. That is the derivative of the original archaeobacterial genome. But the mitochondria have become specialized in energy, the vital force. So it's just like any other city. You have, in fact, specialization with different components doing different jobs. Now, this symbiosis, as you heard from Joe last week, occurred about 2 billion years ago. And as a result now, inside your cell, there are literally thousands of these bacteria, hundreds to thousands. And so there you are a colony. Each cell is a colony of bacteria. Okay? Okay, so how do these bacteria generate energy? Well, they generate energy by burning hydrogen in your food with the oxygen that you're breathing. So when you eat food, food is made of either carbohydrates or fats. And carbohydrates and fats have a lot of carbon and hydrogen. That's how those molecules are made. So inside your body strips the hydrogens from the hydrocarbon and reacts the hydrogen with oxygen. So how does this all work? Well, first, we start with sunlight. Sunlight is impinging on green plants. And green plants also have a symbiotic bacterium. And that symbiotic bacterium is called a chloroplast. In fact, the reason plants are green is that they have this symbiotic bacterium. It's called a cyanobacterium. And that cyanobacterium can take the photon of light and use that to split the hydrogens off of water. So you then get the hydrogen and the oxygen. The oxygen is released into the atmosphere, and that's why there's 21% oxygen in the atmosphere right now. So the hydrogen then is put on carbon, which comes from carbon dioxide in the atmosphere, to make sugar. So in fact, a plant is a polymer of sugar, all linked together. So cellulose is a big long string of sugars. So we then, animals, collect the energy from the sunlight by eating the sugar in the plants called starch. Okay? So the sugar in plants then goes into our mouth, into our guts, into our bloodstream, into our cells, and there the sugar is split into hydrogen and carbon, and the hydrogen then goes to another symbiotic bacteria, the mitochondria, where it reacts with the oxygen to give energy. So all energy flow is not about the nuclear cytosol, it's all about symbiotic bacteria, both in plants and in animals. And all those symbiotic organisms, both the chloroplasts and the mitochondria, came from that original origin of that symbiosis. So all energy flow in complex organisms, plants and animals, all goes back to this original single event that made life, complex life, possible. Okay, so if you don't think that this reaction process is important, then I recommend you just stop breathing for the rest of the lecture. Okay? Okay, so when you burn hydrogen with oxygen, you then convert it into heat or ATP. And the ATP is a chemical structure that allows you to get energy to do work, and heat, of course, maintains your body at 37 degrees centigrade. So how does this all work out from the point of view of DNA? So we really have two life forms. This is the nucleus and its cytoplasm. That's the archaeobacteria. And this is the oxidative bacteria, the mitochondria. Now, the way information flows in biology is information is stored in DNA, which in the nucleus is on chromosomes. The information is converted into a working copy called messenger RNA, which still has the same DNA basis. DNA has a code of A, T, C, and G in different combinations. And the RNA simply makes a copy of that. So it's just like a stenographer that's taking dictation. So the messenger RNA then takes one of the gene's information out into the cytoplasm. And there it's going to be converted into function, because RNA is just a linear molecule like a tape. So how do you convert the tape into a three-dimensional structure like you? You then use the information one unit at a time, which is 3 bases, and that 3 units relates to something called an amino acid. So there are 20 amino acids, and each of them is coded by 3 bases in DNA or RNA. So this process of transforming the linear information in RNA into protein, which is 3-dimensional, occurs during protein synthesis on what are called ribosomes. So there are 20,000 genes in your nucleus. Each one of them is converted into a protein, and that protein then goes from a linear structure into a 3-dimensional structure that then can do a reaction or create hair or be a structure. Okay, so that's how information flows: DNA, RNA, protein. The same system occurs in the mitochondria because it's also a life form. It has DNA, which is circular, not on chromosomes. It makes RNA, and the RNA, again, is read on ribosomes in protein synthesis to make proteins. So what's the difference between the DNA to RNA to protein of the nuclear cytosol versus the DNA to RNA to protein of the mitochondria? The mitochondrial DNA codes for the key enzymes to make energy. And so the mitochondrial DNA is like the wiring diagram of a power plant. The power plant, if you want to build a power plant, you have in the city manager's office big blueprint about where the parking lot is and how many bricks are going to be and what the roof is going to look like. But the wiring diagram of the power plant has to be in the power plant, because if anything's going to go wrong quickly, it's going to be in the electrical flow. And so you have to be close to the energy source to solve that problem. And nature solved that problem in exactly the same way, by putting the energy genes within the energy generating power plant. Okay? So this is then the wiring diagram of the power plant, and then all the genes to build the power plant are there in the nucleus. Okay? So if we want to understand energy flow, then we have to understand this gene.
They didn't have this in high school when I was in high school, so I'm probably...
They didn't have me in high school either.
All right, I understand sort of how within the nuclear DNA, how the base pairs could be separated and replicated. But when they're intertwined and then form a circle, how the heck can that happen?
So the DNA strands are complementary. They're anti-parallel, like so, right? Okay, so this is one strand, this is the other strand, and they're base paired in the middle. Okay, so for a linear DNA, you just pull them apart and make a copy. The circular DNA, you have a topological problem. So what happens is, in fact, you pull the strands apart, and then you make one strand in fragments. Okay, and then you can read around the other strand as a continuous piece, and then you glue the fragments back together to make the circle.
How do we know that this event of fusing two bacteria occurred only once?
Because we can determine the DNA sequence of all the organisms that we're willing to sequence, and then we can deduce what changes would occur. So let's say we have two humans, you two right there, and we sequence your DNA. How many differences are they between you? Well, the number of differences between you is the time since you've had a common ancestor, right? So if we do that for everybody in the whole world, we can go all the way back to their common ancestor. And if we did that for every species in the world, we can go back to their common ancestor. And if we go that every mitochondria and every chloroplast, we can go back to this common ancestor, and there's only one.
Chloroplasts and mitochondria have a common origin?
Yes. The original eukaryotic, so the answer to that is no. The plant cell and the animal cell had a common origin. The chloroplast is one kind of bacterium. The mitochondria is another kind of bacterium. But the first symbiosis was with the cell with the mitochondria. And then later, the chloroplast was added. So a plant has both mitochondria and chloroplasts. But the common origin was with the mitochondria. Now, it turns out that there were multiple different chloroplast origins, and that's why we have brown algae, gold algae, red algae. Those are all different cyanobacterial symbioses. But all green plants had a common cyanobacterial origin, right? Is that clear? Sorry, maybe it was unclear on that.
I just read an article about scientists making the tree of life bigger, and they added like a third branch. And I'm wondering if the first two, if one of those branches was the mitochondria thing, and if you're going down to the bottom of the tree of life, what the heck's there?
So there's a very famous in biology scientist named Carl Woese, W-O-E-S-E, and Carl Woese studied a particular kind of gene that's found in every living organism, called a ribosomal gene. It's one of the RNAs that make up that structure. And he was then able to create a just this kind of phylogenetic tree by comparing the sequences and putting most similar ones closer and the most distant ones farther apart. So when he did that, he found that there was a common origin, but there were 3 branches. What were the 3 branches? Branch 1 was what he already knew were the regular bacteria. We call those eubacteria. Branch 2 was, in fact, what we now know is the genes in the nucleus here. But branch 3 was this other branch, and that became the archaeobacteria. So in fact, the archaeobacteria and the eubacteria formed the symbiosis. And the eubacteria made the mitochondria and the chloroplast. They're still eubacteria. They're just now in a different environment. Okay? And it turned out that the archaeobacteria gave rise to the animal and plant cell nuclei. Okay? So those are the 3 forms of life.
Thank you for joining us. You've been watching Dr. Douglas Wallace discussing how genes are found not just inside the nuclei of our cells, but also inside our mitochondria. For more information on the Origins Science Scholars Program, please visit the Institute's website at origins.case.edu. In the next part of the talk, Dr. Wallace will discuss the variation of mitochondrial DNA across human populations. Now, back to the talk.
What are we going to do today? Why are we talking about origin? We're talking about origin because of a simple experiment that we did a number of years ago. First of all, we did an experiment where we looked at mother and father and children, and we looked at the DNA inside the mitochondria, and we looked at variation. And what did we find? We found that contrary to what we were taught in school, where mother and father each gave 1 gene to their offspring, and since the mother and father each had 2, each offspring could have different combinations. What we found is every child had exactly the same mitochondrial DNA as the mother and never the father. Okay? So we showed that the mitochondrial DNA was transmitted from mother to all of her children and her daughters from their children, but male mitochondria enter the egg, are seen as foreign, and are selectively destroyed. So men have been thrown out for 2 billion years. It's really appalling. But as soon as we realized that, then we realized that if I sequence the mitochondrial DNA of everybody in this room, the number of nucleotide differences between any 2 of you would be proportional to the time you had a common mother. And therefore, if we sequenced everybody in the room, we could literally reconstruct the maternal lineage that brought you all together. Okay? Or even broader, if we looked at Aboriginal populations from around the world, we could find out what the common mother was. And knowing where all those populations were and knowing their genetic relationship, we could in fact reconstruct the origin and migration of women. Now, those of you that have a Y chromosome will be reassured that we also did the Y chromosome study, and we found that we went more or less together, which I suppose is not so surprising given the divorce rate. But anyway, so this tree, which we'll come back to, is actually a diagram of how our ancestors started in Africa and migrated out to colonize all the rest of the world. And we'll come back to that. But that's really, from my point of view, old news. That's work we did in the 1980s. What surprised us and everybody else is that these mitochondrial DNA types, which we'll get back into, are highly geographically oriented. And that is totally different from your nuclear genetic variation, because every population around the world shares many of the same nuclear variants. But in fact, the mitochondrial DNA variants are highly geographically and constrained by individual population. So the surprising thing is the question why. Okay? So why is it that there was a single origin for human mitochondrial DNA? It doesn't mean that there was only one woman, even though that would be a biblical idea. No, there were probably at least 5,000 to 10,000 women, but only one type of mitochondrial DNA started the whole process. Why did that happen? Or moreover, why did only 2 mitochondrial DNAs, of all the thousands that lived in Africa, only 2 mitochondrial DNAs left Africa and colonized the rest of the world 65,000 years ago? Or why, of all the mitochondrial DNAs in Eurasia, only 3 mitochondrial DNAs became concentrated in in Chukotka and crossed to colonize the Americas? Why were these huge bottlenecks in mitochondrial variation? And that has been the question I think has been preoccupying me for the last 25 years. Okay, so we're going to talk about that. So we're going to first ask, why maternal inheritance? Why would it be important to have one mitochondrial DNA? So here what we have is we've taken a mouse and we've generated its embryos, and those embryos have the ability to make a whole mouse because those are embryonic stem cells. So we can take these embryonic stem cells with the mitochondrial DNA from that mouse. We can then take another cell with a different mitochondrial DNA. This is 129 mitochondrial DNA, it's just a number, NZB. We can take this cell, remove its nucleus, take this cytoplasm with this mitochondrial DNA, and fuse it to this cell. So we've now mixed the two mitochondrial DNAs. We can put that pluripotent stem cell into a blastocyst, put it into a foster mother. She's kind of embarrassed. She's gotten, anyway, and then breed the female to see if we can get a chimera, which will be some of these cells plus these cells. And so these are brown, this is black. And then we can take this female, and if the brown cells made her eggs, then we will transmit the two mitochondrial DNAs into the mouse. Okay? So what we can do then is we can then artificially mix these two mitochondrial DNAs, which would never happen in evolution, in normal life, right? Because of maternal inheritance. So what happens? Here's an example. This is the founder mother. This is the one mitochondrial DNA, and this is the other. So you can see that she has the two different mitochondrial DNAs. Her daughter, this is females, this is a male, these are a mating. Her daughter has the two mitochondrial DNAs, and when this daughter is mated with this male, all of her offspring have the two mitochondrial DNAs, and all the daughters have the two mitochondrial DNAs. But the males with the two mitochondrial DNAs mated with females with only one, they only get the one, showing maternal inheritance, okay? There's absolute maternal inheritance. But we're transmitting this mixture, so-called heteroplasmy, through the maternal lineage. Never happens in normal life. So what happens? So here is an animal with a uniform nucleus. We then have the mixed heteroplasmy, and we then segregate out to an animal that has one type, an animal that has the other type, and we're going to compare the three to see if there's any difference. Remember, the nucleus is absolutely the same. Okay? So if we then look at the activity of a mouse, and a mouse is active at night, then this is the 129, pure 129 mitochondrial DNA, active in daytime, low, high active at night. This is the NZB, active at night, low at day, active at night. But look at the heteroplasmic animal with the two mitochondrial DNAs. It's hypoactive, it's depressed. If we now put that animal in a stressful environment, it's completely uncontrollable. It actually goes crazy. So it's manic depressive. It has a manic depressive phenotype. If we then ask that animal to learn a task, so we're now going to put the mouse in the middle of this field. We have red, green, blue, and yellow, so it can orient. And behind this little hole, there are holes all the way around this circle, is a little black box, and a mouse wants to hide. So we put the mouse in there, and it wanders around until it finds the hole and jumps in. And then we can time it, okay? So if we look at the 129 animal, this is the first day it got tested, the second, the third, the fourth. It learns where the hole is because of the colors and goes in quickly. And the NZB does too. The heteroplasmic animal learns slower. Okay, that's fine. But now we just let the animals rest for 24 hours. Just don't show them this experiment again. Now we put the 129 animal back in the case, back on the plate, and it runs and jumps in the hole. We put the NZB animal on the plate, runs and jumps in the hole. But we put the heteroplasmic animal in, and it has absolutely no recollection that it ever saw this before. So by simply mixing two perfectly normal mitochondrial DNAs, we have a severe manic depressive phenotype and a massive lack of long-term memory. And that's why there's uniparental inheritance. You cannot mix two wiring diagrams for the same power plant or the whole thing shorts out. Simple physics, okay? All right, so this is the wiring diagram. It codes for 13 proteins that are necessary to make energy and the RNAs for the ribosomes to convert the message into protein. And there are many mutations in this circle that give you genetic diseases. But there are also variants that are very ancient. This variant in the circle of mitochondrial DNA is found in 3/4 of sub-Saharan Africans. This variant is found in about half of Europeans. Variant A, B, C, and D arose in Central Asia, and they crossed the Bering Land Bridge to colonize the Americas. I'm going to argue that these variants allowed our ancestors to adapt to the different environments they found as they moved around the world. And it's that adaptation that makes those mitochondria stick to where they are, because that's where they're good at being, right? Okay. So again, mitochondrial DNA is maternally inherited. There are thousands of mitochondria in each cell, and they're constantly replicating inside your cells. So they're growing, and then they're being eaten up by a process called mitophagy. So it's an active colony. In fact, if you don't really believe that this is true, lie awake tonight when it's really dark, put on your little mask, and if your maid is not snoring, that's really distracting. If they're not snoring, you lie there very quietly and think about it. You'll feel them wiggling in there. Anyway, the point is that as they're replicating, they accumulate damage. And so over time, the number of mutant mitochondrial DNAs accumulates, and that's aging. Because as the mutant mitochondrial DNAs increases, the energy output of the system declines. And when I asked my mother what it meant to be old, she said, I just don't have the energy I used to. I didn't believe her until now I'm 70, and now I can hardly get up the stairs. So this is, in fact, the process of aging, decline in energy. So as the amount of damaged mitochondrial DNA increases, it ultimately falls below the minimum for that organ to function. And the most energy demanding organ is your brain. It's 2% of your body weight. It uses 20% of the energy. Therefore, a 5% reduction in your mitochondria is more than enough to give you a massive headache. And so as you'll see, that is in fact the cause of all the common diseases, variation in energetics. Okay, so here's an example. This is a family. That's a father, that's a mother, these are their children, and these then are their marriages. And this woman had a mutation in her mitochondrial DNA in a gene called ND6, and it just changed this protein, amino acid proline at amino acid 25 to a leucine. This woman was 50% heteroplasmic. 50% of the mitochondrial DNA had the mutant, 50% did not. And she had problems with her eyes and her balance. Her sister only had 5% mutant mitochondrial DNA because it's randomly distributed through the maternal lineage. But she had 3 different consorts, and every one of her children had 100% mutant, and they all died in childhood. So this extranuclear DNA is not inherited by the rules of Mendel. It's inherited randomly by a stochastic process along the maternal lineage. And the more mutant, the worse off you are. Okay? So we can make a mouse. We have exactly the same change. ND6, proline 25 to leucine. We put this mutant mitochondria into the mouse female germline. So what do we get? This is the optic nerve of a normal mouse. This is the mutant mouse. And you can see that the axons are swollen. They've lost the myelin. And we get a selective loss of the high energy neurons. And these animals go on to get a neurodegenerative disease. This is another animal that we created. Here we have an animal that has 2 different mitochondrial DNA mutations, one in the ND6 gene, but this is a very severe one. It kills that gene. I'm sorry, I should have mentioned. This mutation, this mutation is only causes a 30% reduction in energy. But this mutation, if it were pure mutant, there would be no energy. But that's not the case here. This is a mixture of this mutation that's very severe and this mild mutation. We're going to put that into a mouse. Okay, so what do we get? Well, here is then the tissues of the derived mouse. And the blue is just the mild mutation, and the red is the severe mutation. It is heteroplasmic in different organs, okay? So then we take that animal, and it has about 50% of the severe mutation, and we cross her with a normal male. And what we found is all of the pups, all of the offspring have, in fact, instead of 50%, they have 16% of the mutant. And then we cross her again. The next litter is only 6%, and then the 4th, 5th, and 6th pregnancies are no mutant. So there's a directional elimination of the most severe mutation through the female germline. Okay? Why is that important? Because the mitochondrial DNA has a very high mutation rate, and most mutations are bad. So if, in fact, all the mutations were put into the population with a very high mutation rate, it would kill off the species. So what happened? Mother Nature built into the female germline a way of testing each egg. And if the egg had a really bad mitochondria, it killed it off and didn't allow that to contribute. So what does this mean? This means that all of us mammals, half of all of us mammals, all of you female mammals have a very high mutation rate for your mitochondrial DNA, but you're only allowing the best variants to go out into the population. The bad ones you're throwing out. And that allows a lot of variation to exist in the population. Okay? So almost everybody in this room has a slightly different mitochondrial DNA and a slightly different energy metabolism. Okay? Because of this high mutation rate.
I'll see if I can phrase this in a way that makes sense. But so when these mutants appear, when they're inherited, is there anything that determines which is dominant, or is it just the fact that they're there and they screw up the wiring?
Yes and yes. So what happens is, first of all, the egg, the female egg, has about 500 to 1 million mitochondrial DNAs. Okay? So inside that egg, you might get 1 mutant mitochondrial DNA. All the rest would be normal, right? But then when that egg gets fertilized with a sperm, it starts dividing into making cells to make the blastocyst and then to go on to give the animal. Well, up for the first large number of cell divisions, it doesn't replicate its mitochondrial DNA molecule. So what it does is it replicates the nucleus, but not the mitochondrial DNA. So literally you take that 500,000 molecules and then randomly divide them up on all the cells. Okay? So just a small number. It turns out about 100. Then only a few cells actually give rise to the female germline. So what you've done is you've made a sampling of a very large number of mixed population. And that, by sampling a small number, you'll create a large statistical variance. That is, you either get a bad one and then you have high mutant, or you only get good ones and then you have normal. And so by this sampling error inside the germline, you create very bad eggs, and I mean very good eggs and very bad eggs. The bad eggs then, when they start to grow, they have an energy deficiency, and as they start to ovulate, they die. And the good eggs are ovulated and can get fertilized. So you have a system of sampling error, statistical sampling error, giving you large distribution, followed by selection to get rid of the bad ones, and that preserves the genotype. But if the mutation is mild or beneficial, then that will rapidly become an egg that then gives rise to a new adaptive lineage of the genome. And that's the key idea here.
We hope you've been enjoying the Origin Science Scholars Program with Dr. Douglas Wallace. Dr. Wallace is director of the Center for Mitochondrial and Epigenomic Medicine at Children's Hospital of Philadelphia. In the second part of our talk, we learned how variations in human mitochondrial DNA emerged and were carried around the world. In our final segment, Dr. Wallace will discuss the mitochondrial basis of disease. Now, back to our talk.
So this just shows that the milder mitochondrial DNA mutation will give you, in this particular mouse, we've now lost the severe one because it's segregated, but we now have the mild one, and this mild one gives you a muscle problem, a heart problem, and diabetes. So nucleus is normal, but a single point mutation in the mitochondrial DNA has already given you all the complex diseases. Okay, so this is a human family. These people have all gone blind. They're all related through the maternal lineage. But note there's 4 times more males that are affected than females, and all of them get this exact same ND4 mutation. So why is that interesting? Because this sex bias is seen in Parkinson's disease, it's seen in autism, it's seen in many different genetic diseases, and we feel this is characteristic of milder mitochondrial disease. But there's also another factor, and that is, even so, not everybody, not all the males are affected. So why is that? Okay, so this is a disease where you're fine in normal life, and then suddenly in midlife you lose vision in one eye and then in the other, and are legally blind, called Leber's hereditary optic neuropathy. It's a maternally inherited mitochondrial predisposition. There are multiple different mutations in the mitochondrial DNA that will give that blindness. This is a severe mutation, this is an intermediate one, and this is a mild one. That's the biochemical defect. So if they have different biochemical defects, why do they give the same problem? If it was really a quantitative thing, you'd think a milder one would be less affected, right? Well, the reason is because this isn't the only factor. it depends on what the mitochondrial DNA that the mutation occurred was like. So this is a lineage we're going to call J. It's found in 10% of Europeans. This severe mutation, it will give you blindness whether you're a J or not. This is just random sampling. But this milder mutation will give you blindness if you're also J. That is, you have a higher probability. And this very mild one will almost have to be J to give you blindness. There are other mutations like this 3394 variant. So it's not just a single nucleotide change, but also the maternal lineage on which the change occurred. Okay? So that's back to this. So remember what we did here is we went all over the world, found Aboriginal populations that had lived there for 10,000 years, sampled them, did a very careful anthropological study, and then sequenced their mitochondrial DNAs. And then we could build together this whole phylogenetic tree. So these are the Khoisan people of the Kalahari. These are two different African pygmy lineages. You think pygmies look the same, but this one has existed for 90,000 years, and this is only 30,000 years. Okay? Still the same adaptation. This variant, sub-Saharan African, gave rise then to all the people up here in Ethiopia. Two mitochondrial DNAs arose, M and N, 65,000 years ago. Of all the hundreds to thousands of different mitochondrial DNAs, only two maternal lineages left Africa and colonized the rest of the world. One lineage, M, stayed in the tropics all the way down to Australia and much later moved into the temperate zone to give lineages we called C, D, and G. These are just letters as we discovered them. But N moved directly into the temperate zone and gave these European lineages, H, J, T, U, U, K, V, I, W, X. These, of course, have been most studied because there's the most money to study them. Okay. Then much later, one of the lineages from this N lineage called A, and two of the lineages later from the M lineage, C and D, became enriched in Chukotka, and they crossed the Bering Land Bridge to colonize the Americas. Okay? So this is a huge geographic separation of these types. So why would that be? Because here in Africa, if you're a woman and you want to reproduce, you need to be able to run away from lions. Being run away from lions, you need to have very efficient mitochondria. You have to take all the calories that you eat and convert them into ATP, because that's how you run, right? You heard that from Joe. And it's hot, so you don't want to make a lot of heat. Well, a calorie is a unit of heat. Every calorie you burn, you get that amount of heat in your body, okay? So you want to minimize the number of calories, maximize the number of heat to run away from lions. Okay, that's fine. But up here, the lions froze to death. That wasn't a problem for you. The problem is you froze to death. Okay, so how did the women survive up there? They changed their efficiency of their mitochondria. So now they were less efficient. They had to eat more calories for the same amount of ATP, but burning more calories generated more heat. What was going to kill them was the cold. So it was better to be inefficient to make more heat. Okay? So changing the energetic efficiency will allow our ancestors to adapt to different environments. Now that meant that these people had to eat a high-fat diet because fat has a lot of calories. So they had to kill marine animals, and we all looked down on them. I'm sorry, that's too bad. That's a niche. So this, in fact, then, was how our ancestors actually came to live in those different places. And therefore, why is there only one mitochondrial DNA in Africa? Because a small population of proto-humans happened to become isolated in a particular part of Africa with a particular mitochondrial DNA that was good for that area. And that then gave rise to all the rest of the variation of our species. Okay? It wasn't that there was Adam and Eve. It was that selection limited the amount of variation in that environment. Okay? Is that clear? Because that's a key idea. Yes, sir. Did you have a question?
If this happened to humans, if this happened just
I'll repeat it.
or other,
He says, did this happen to just to humans? No, I believe this is the missing evolutionary principle that makes neo-Darwinian work. Okay? And that's what I'm going to come back to. But our studies on human medicine gave us the insight because we looked enough within a species to figure out what variation really meant. It's not about whether we have an extra arm or not. It's whether we are cold in the winter or hot in the winter. Whether I'm fighting with my wife because she's Scandinavian and she's always hot, and I'm not Scandinavian, I'm always cold, and we can never agree on what the thermostat should be. Okay? That is mitochondria in action. That is divorce. Okay. Anyway, this is an example coming from just Europe. 10 to 15,000 years ago, this transfer RNA had a single change, an A to a G. That A to a G is a very minor change. It didn't affect the reproductive capacity of human women. But what happens if people live long enough, it certainly affects the quality of their brains. So this variant is only about 4% in people that are young. But in people with Alzheimer's and Parkinson's disease, it's 7%. So this is a mutation that has no selective disadvantage because evolution only cares about reproduction. But because we wish to live to 100, this then becomes deleterious. It's called antagonistic pleiotropy. Very classic idea in evolutionary biology. And we see this over and over and over. This is another mutation that is much more severe, and it will give you Alzheimer's and Parkinson's disease if you just get it. You don't have to keep that thousands of years to get that mutation. Okay, so here is another variant. This variant is, this is the lineage of M out of Africa. This is the N lineage out of Africa. And these are mitochondrial DNAs we sequenced from Tibetans in high altitude. And you can see on the M lineage, we have this 3394C mutation occurring 3 times. And if we look at the frequency of 3394C versus altitude of the village we studied, it's clear that there is strong selection for this mutation. So this mutation changes energy metabolism to deal with low oxygen tension. Okay? Now what's amazing about this 3394C, this is good at high altitude, but this is one of the same mutations that at sea level increases the penetrance of labors. Here's another case of antagonistic pleiotropy, but now the difference is altitude, high or low, as opposed to young versus old. Okay? So in fact, there is a whole bunch of associations between the different mitochondrial lineages and disease. And we've correlated them with Alzheimer's, Parkinson's, macular degeneration, migraine, psychiatric disorder, stroke, diabetes, cardiovascular disease, metabolic syndrome, sepsis, IgE, asthma, AIDS, therapy, osteoarthritis, altitude adaptation, aging, cancer, and athletic performance. Now this is my most important observation, because when I was a boy, my father, he was a big advocate that all boys should be good baseball players. Okay, so I was his son, he dragged me out, he coached Little League, he was such a noble father. And I was an unmitigated disaster. So I'd put it out in the outfield, the ball would come, and I'd run around and hit me in the head, and he would go, oh God. So he put me as a catcher, I'd be down and it would hit me in the face. And finally, he was completely fed up. And I felt bad about this, you know, I've always felt bad that I disappointed my dad. Well, I thought, you know, it was just some kind of incompetence. But then I began to learn about mitochondrial DNA lineages. And one of the things Dad said was, well, if you can't play baseball, why don't you run long distance? I said, well, why would I run long distance? Nobody will ever see you out there. Okay. So, okay, I tried to do that. It was humiliating because I was always last. I could figure out, I trained and I ran and I was always last. So he said, well, don't do that anymore anyway. So you know, I did dumb things like I would do a Boston Marathon. I'm going to show him. I couldn't finish. Who finishes the Boston Marathon? These Ethiopians. I mean, the Kenyans. Damn it, the Kenyans always win the Boston Marathon. In fact, they win all the long distance races. This is so humiliating. Why would they win the long distance races? And then I learned that, in fact, if we look at mitochondrial lineages, these are the European lineages, these are the African lineages, and lineage L0 is the most efficient mitochondrial DNA. And all Kenyan long-distance runners are L0, okay? And I'm T. Holy smoke! I suddenly feel much better. I now know why I could never run long distance. I feel better about my father, but now I hate my mother. Okay, so there are a huge number of phenotypes that are related to mitochondrial variation, and they're exactly the ones you care about. You don't care about whether you're going to have an extra arm or not. That's something about deep evolution. What you care about is whether you're depressed, or whether you're going to get macular degeneration, or whether you're going to get neurodegenerative disease. And people have looked and looked and looked in the nucleus, and if you follow the field, we can only account for 10 or 15% of the variance from all the variation around the world for these diseases. And they say, where's the missing variance? You know what? They never, ever once looked at the mitochondrial DNA because it was irrelevant because they believed in Mendel. Okay? But if we look at mice with these different mitochondrial DNA variation, and we just look at something simple like a neuropsychiatric response, okay? So we're going to do something equivalent to my being told I have to write a grant. That's not the same as actually spending the month sweating blood and tears and writing a grant. I'm just being told. So we're going to take the mouse and we're going to put it in a little confined environment, like a hole, for 30 minutes and then let it out. I mean, it's nothing, right? Nothing. What happens to this mouse? So here we're looking at it. Here's the mitochondria. These are the enzymes, 1, 3, 4, and 5. This is a gene that exchanges the ATP, and this is an antioxidant gene necessary for the mitochondria. So we have all of these mice, and we're going to expose them to 30 minutes stress, and we're going to ask what happens to their stress response. So when you get stressed, you make corticosterone. This is a kind of stress hormone, okay? So we should see then what happens for corticosterone when we stress these mice. So this is the mutation in the ND6 gene. This is the mutation in the CO1 gene. And you can note that the ND6 gene, the corticosterone for the 30 minutes of stress goes up, and it stays high relative to the CO1 and the wild type. If we look at glucose metabolism, which should be downstream of glucocorticoids, we see the ND6 goes up and then down, where it's much less for the wild type. But now let's look at the nuclear genes. This is the ANT that exchanges the ATP and ADP. Look at the huge effect it has on glucocorticosterone and then down, yet its ability to change glucose metabolism is null. On the other hand, the antioxidant mutation of the mitochondrial DNA has no effect on glucocorticosterone, but a huge effect on glucose. What am I saying here? These are the things your psychiatrist would look at if you were stressed. And he would say, well, glucocorticosterone is very important, we'll measure that. But I'm showing you the same stressor has totally different effects by very subtle mitochondrial changes, okay? So if we then just compare the ND6 and the ANT, and we look at the change in glucose, change in glucocorticosterone for a 30-minute stressor, we can see that they're on totally different sides of the stress response. So that's why people here are going to differentially respond to stress, because you all have a different mitochondrial genotype from the mitochondrial DNA. And as soon as you're put into a stressor environment, you're going to have these kinds of differential responses, okay? So let's look at something you might have heard of. These are so-called catecholamines. Dopamine, norepinephrine, and epinephrine. They're the stress, flight, fight hormones. So here what we're going to do is put the mouse, normal mouse, and put it in the little 30-minute constraint. And this is one genotype, one genotype, one genotype, one genotype. So dopamine, they all show an increase in dopamine. But now look at norepinephrine. These are up, but this one is already so stressed out, it doesn't even respond, okay? And now look at this epinephrine. These are still responding, this is less, but this one is completely wiped out. Again, these mice look perfectly normal, just as normal as you do. But a subtle change in energy metabolism has completely changed the psychiatry of that mouse, okay? And this just shows that there's something called inflammatory stress. This ND6 mouse shows inflammation, the NNT shows inflammation, but the other three have less inflammation. Well, that's asthma and inflammatory responses to all kinds of things, okay? And if we just look at gene expression, the mitochondrial DNA mutations are totally different from the nuclear mutations. The point I'm trying to say is there's a huge amount of variation in mitochondrial DNA just among this population. It didn't affect the fact that you could walk here, or that you walk home, or you eat dinner, or any of those things, but it radically affects how you think about yourself, whether you're predisposed to disease, and to all of the other clinical things that are important about being human. Okay, just one more point. So the idea is that we have two genetic systems, the mitochondrial genetic system that's important in energy, and the nuclear cytosolic system that's important in anatomy. But in medicine, we're not worried about anatomy, because if you're alive, you have a normal anatomy. So therefore, we must be worried about energy. And yet, 98% of all the federal budget has gone into the nuclear cytosol and almost nothing in studying the mitochondria. All the association studies on common diseases have studied only the nuclear DNA and never the mitochondrial DNA. So if we don't ask the right question, how can we get the right answer? Okay? So the last idea is that the mitochondria then is your sensor to the environment. There are all kinds of environmental challenges. Your mitochondria perceives those by changing energy. It sends a whole series of signals to the nucleus, and the nucleus then responds by changing the gene expression to reconstitute normal function of the mitochondria. And as long as we can maintain that, we have energetic homeostasis and health. But if there's a defect in energetics or the environment, then in fact we get energetic decline and disease and death. So, if we now put energetics at the center of medicine instead of anatomy, then all of the genetic diseases have the same origin. We can have genetic changes in the nucleus that will affect energy, mitochondrial changes that affect energy, but also calories and how you use them will affect energy. If energy is defective, you will damage your mitochondrial DNA. That gives you age-related decline in energetics, and that's aging. And a minor change in energetics will affect the most energetic tissues, the brain, heart, muscle, and renal. Those are all the common disease organs. And if you foul up the power plant, then you won't burn the fuel. That's glucose and fatty acids, and that's why diabetes has high levels of glucose. And finally, inflammation is all about energetics, and the mitochondria are bacteria. So if the bacteria are released to the bloodstream, you're going to get inflammation. And that's why inflammation is present in all the common diseases. And finally, cancer is all about cells having enough energy to grow. So if we just stop thinking about energy, anatomy, and start thinking about energy, then we have a single cause for all common complex diseases. And that means we could treat them, because all we have to do is treat energy. Why don't we do that? Pharmaceutical industries are not interested because they believe one gene, one polypeptide, one disease, and those genes are nuclear. So I'd just like to finish by thanking all the great people that have done this work. Dimitra Ashaklia was very important in the population studies. Megan, Martin, and Ryan Morrow were very important in the mouse studies. And then we have studies on Tibet with Fujian Yi and my colleagues, Lorna Moore and others in Chongqing and in Tibet. Studies on neurology with my colleagues at CHOP. Cardiological studies with Jargat Narula, and a whole series of ophthalmological studies, and of course, some support from NIH. Thank you very much.
The Origin Science Scholars Lectures are presented by Case Western Reserve University's Institute for the Science of Origins, with the assistance of the Siegel Lifelong Learning Program, the College of Arts and Sciences, and MediaVision. For more information on the Origin Science Scholars Program, including a full video archive, please visit the Institute's website at origins.case.edu.